biology. She's a lung immunologist throughout her career. She's now the Associate Dean for Graduate Student and Postdoctoral Affairs, I think it's called. But she looks after the good experiences of graduate students and postdocs and addresses all of these kind of research integrity kind of questions. Second part of the series, the session today will be Carrie Oliver, and I'll introduce her when she comes, but she's in the office of the Institutional Review Board, the IRB. Okay, Lisa, thank you very much.
All right, let's see if I can. Good morning, everybody. Let's see if I can not electrocute myself here. That would be bad. Okay. Thank you all for coming, and thank you, David, for that introduction. So as you gather, I wear a bunch of different hats. I've been here now, I can say officially, more than 20 years, and have really touched upon a bunch of different experiences which have led me to this particular job, I guess, one of the hats that I wear. For those of you who has attended one of my RCR workshops, because I see a few familiar faces. Okay, so you know all the punchlines, so don't give it away. Okay. But again, too, thanks for those who are out in the ether and remoting in through GoToMeeting. And please, this is to be informal. Ask questions, interrupt me as needed. Certainly, I have a ton of information in here, more than we'll get through in the next 45 or 50 minutes or so. But Jean's going to send out all of the slides with embedded links because there are different activities that you may find interesting and frankly, informative and fun, I guess, in a really nerdy kind of way. I think it's fun. All right, so let's dig in. So you may have glimpsed from setting up that responsible conduct of research. What does that all mean to you? So this is where we don't move on until somebody says something. Wake up. What is responsible conduct? Ethical research, what does that mean? What are ethics, I guess?
Trying to protect your participants.
Okay.
They're right.
Okay. So yeah, looking at it from a perspective of clinical science, translational science, very much focused on IRB, which you'll hear more about in the second hour. How about integrity? What's integrity?
So one thing is to focus on scientific rigor.
Okay.
Because studies are done in a rigorous fashion.
So I bet you've completed the Kaizen platform thing. But you're right, that's part of it. So integrity is doing the right thing when no one's looking, right? It's where your values and your actions intersect. And you do the right thing, again, when no one's looking. So that's what we're going to talk about a lot today in the context of RCR, excuse me, and science in particular. So scientific integrity is public trust, right? We do a lot to impact society. And in fact, they support our science through taxes, participating in clinical trials, studies, and really relies on it to impact their lives. So can you give me some examples of how you think science has impacted society? Vaccinations, especially in flu season. It's a good answer. So right, that's one great example. So today, we're really thinking of this as a refresher session because you may have touched upon this in other areas as well. And again, you'll receive the slides. But I want to review the guidelines, certainly definitions, overall structure of RCR. But importantly, too, we'll have time to work through a couple of case studies, I'm hoping, that can maybe give you skills and tools to use in your own labs or clinics that can impact your day-to-day. So if you go to the NIH website and look up their Office of Research Integrity, you'll see a whole list of resources and items to dig into. And that's a lot of where I have gathered information for today's talk. But really, it's focused on these 9 different areas. Human subjects, certainly, again, you're going to hear about later. Peer review, collaborative science, all that you see up there. We're going to talk in somewhat a little bit detail of data management and authorship. But for me, really, bless you, mentoring runs through all of this. Whether you are a mentor or you're certainly working with a mentor, your actions, as a role model, can really impact those that you train with or train under you. So what is research? Bless you. What is research?
Learning for outcome.
Trying to move the needle, right, in a field. So that really systematic investigation to develop that knowledge, again, to move the field, to move the needle, to impact society. So through research, we're always generating data, yes, to have that impact. So we generate the data, that builds our knowledge, and ultimately leads to that impact action, maybe developing new vaccines, developing new models for research and study. But what are data? We throw that term around a lot. What are data?
Information.
Information. What kind of information? Numbers, audio. Okay. How do you get those numbers? Yes, exactly. But I guess, what are the tools? Right.
You can use surveys.
Okay. So survey is a tool. Is that data? The survey? Mm-hmm. Why not? It's an instrument.
It's an instrument to
Okay. How is that calibrated? Mm-hmm. Data are everything from start to finish. Everything from the protocol and methods, the actual tools that you use, how they're calibrated, how they're used, or like for a survey, has it been vetted? Has it been tested? Also, the analysis, the statistical approach that you use, that's data. So when you think of fabricating or falsifying data, it's everything in that stream. What if you were not to calibrate a particular piece of equipment? Can you trust the numbers that it spits out? No. So that too are data from start to finish. So to again think about what we're talking through in that realm. So research misconduct, overall, you may have heard the acronym FFP. It's falsification, fabrication, and plagiarism. So I put up these definitions from different sources. Oh, this is where I get to use the new tool. Maybe. There we go. From different groups, different institutes, to show you that it really does cross discipline. So I facilitate RCR not only for School of Medicine and related biomedical efforts, but also across campus. And really their perspectives of RCR, for example, in computer science, albeit that's a very different area, there are threads that run throughout the same. Mainly calibrating tools and instruments and making sure that they are validated for proper use. But again, I show these to show that commonality. UAB goes one more step. If you have dived at all into any of the policies for conduct, it's also intentionally withholding information about a case, certainly bringing malicious charges about misconduct against someone. And we'll talk a little bit later about that process, how that all happens and unfolds here at UAB specifically. But also certainly any interference or retaliation of an ongoing investigation. So it's not only that potential misconduct itself, but everything that surrounds it in terms of interactions and behavior. So what's fabrication? We know that misconduct is fabrication, falsification, and plagiarism. So what's fabrication?
So people off-site are
Yes.
not able to hear the audience answers, so if you can repeat them, that would be great.
Okay, I can do that. So fabrication, the creation of false data, so making things up. Yes. Again, looking at the definitions across disciplines, it's shared. So making up data or results. So again, that term of data, making up numbers, making up your own analytical tools that haven't been vetted, for example, things in that realm. So just an example in that regard. This is a pretty old one, but it illustrates the point fairly well. So who you see here is Eric Pullman. He was a researcher at UVM up in Burlington, Vermont, a number of years ago, and had generated work that ultimately was awarded over $500,000 in federal grant monies from the NIH. Well, turns out he had fabricated his data. There was a whistleblower who turned him in. Office of Research Integrity conducted its investigation, and sure enough, they found him guilty. He is noted for being the first scientist to commit fraud and receive jail time. He served a year and a day, as well as fines, and certainly was banned from ever receiving any other federal money. Guess what he's doing now? Just take a guess. He's actually a college counselor in Montreal. I have 2 teenagers. That kind of hits home for me. This guy is, yeah. Never mind. So that's a pretty infamous example. There are certainly others. So if fabrication is making things up, what's falsification? Changing things, tweaking it a little bit, maybe calibrating that instrument in a way that's going to give you the numbers that you want or are correct, and I'm doing air quotes for those online. So right, it's a little different. Manipulating the data, the numbers, the materials, the equipment or so forth, such that it's going to generate information that's not accurate, right? No alternative facts here, no. Another example, this one, more recent, 2015, Akil Pody. You may have heard of this example or researcher. He was at Duke, and like the former example, had garnered a good deal of NIH monies. And another whistleblower status, these were biostatisticians who, looking through or combing through his work, found that there was falsification of data. This ended up certainly in his being dismissed from Duke, banned from getting any other federal monies, also to retraction of nearly 20 publications. Who does this impact besides him directly, as well as the other example? The human subjects. Take that one more step. Who else? The university.
The research community.
How does it impact the research community? It's just his work.
Oh, because lots of base there.
Right. Exactly right. So others that go ahead and read through the literature like we all do to base our next experiments on what's been published, trust that this is done correctly, right? So it impacts certainly the subjects, the research community, society on the whole. He took away money from others that could have been funded, in days of tough funding, like now and then. So it does really have a larger ripple beyond just his own group. Guess where he is now? No, he's in Nebraska, seeing patients.
He's in where?
Nebraska,
Treating?
treating patients. He's not doing research, but he's treating patients. Just putting that out there. Here's another example. You may also have heard of out at Ohio State. Carlo Croce, very famous cancer scientist. Never want to be published like this in the New York Times, I don't think, though. Let's see if this works. I want to see. We can get the internet going here. So he is at Ohio State, as I say. A number of different allegations have been made about him over the years. And Ohio State has essentially kind of just not looked into it largely. Scrolling down, okay. So the allegations that have been made against him have certainly been not only plagiarism. There's been an allegation that he plagiarized a junior colleague's grant, took whole sections of that grant and submitted it as his own to the NIH. And then this. Let's see if we can get this to work. So also duplicating messing with gels. So essentially copying and pasting to get the picture that he wanted, making it pretty, right? So he's still doing research. Member of the National Academy, and I believe he is still that. So some pretty famous examples, not to be left out, I guess. There has been a famous example here at UAB several years back. Never want to end up on the front page of Birmingham News like this. This was a study out of the transplant group here, looking at the impact of an anti-rejection drug. So they were using primates, looking at transplanted kidneys. So essentially, the design was to remove both kidneys from the primate, transplant one back, give them the drug, and then monitor kidney function. So this was the protocol, as I say, going through. The primates were having wonderful, almost normal kidney function in the midst of this drug until there was a whistleblower. There was a trainee who blew the whistle and said, well, in fact, they didn't remove both kidneys. They only removed one. So autopsy on these primates showed that the transplanted kidney actually was not functioning at all. It had completely necrosed. And the primate was working off of its own original kidney. Yeah.
Were the researchers receiving funding from a pharmaceutical?
So the question was, were the researchers receiving funding? They were receiving NIH funding that had to be paid back by the university. There was a 2-year investigation. They were ultimately dismissed. The lead investigator was banned from receiving any more federal monies. They've certainly left the UAB, as I mentioned. Yep. So again, society is impacted. The university is impacted. Who else is impacted? Getting a little more granular.
I think just see something like this and then they don't want to participate in clinical trial.
Absolutely. Absolutely. It's going to hamper potentially future trials. But look around the room. Exactly. The trainees. So those impacted include graduate students, medical students, postdocs, technicians. Technicians lost their jobs. Trainees had to find other labs or other work or labs to work in. So even within here, it impacts. And this is our last example that we'll wrestle with, but I think probably nothing better shows the impact of fabrication and falsification on society writ large. As I'm sure you're familiar, the MMR vaccine and the autism debate that's sadly being still debated, despite the fact that this study was very small, loaded with errors and misconduct, fabricating data, falsifying data, you name it. And still, it's an ongoing debate that's been picked up by society in the conversation and now, impacting herd immunity, again, writ large. So we've talked about F and F. Let's now talk about the P, the plagiarism. So what's plagiarism?
Copying or taking work that's not your own and putting your name to it.
Right, exactly right. I'm sorry. So plagiarism, as shown here, the appropriation of another person's ideas without giving proper credit, right? So sometimes you'll see a type of plagiarism wherein there are only, it's called mosaic plagiarism, wherein you can see that the article on the right puts in a few different words here and there to make it different, right? So microRNAs, or the miRNAs, whoops, back, there we go. And again, a few words here and there. Why, if you put this through Turnitin or iAuthenticate, would it be picked up as being plagiarized? Why not?
It's not an exact match.
Yeah.
I taught, so I would have to read it for myself to make a final decision. It's not an exact.
Right, but the layering of the concepts is still the same. Right, exactly right. Is this plagiarism? 87 years ago, our ancestors established on the North American continent a new country based on freedom and aimed at the big idea that everybody is just as good as everybody else. Now we're struggling against each other in a contest of whether this country or anyone like it can survive for very long. We stand here where we have fought a bloody battle in an effort to convert part of the area into a sacred place for those who made the ultimate sacrifice for the country. It's a noble thing to do. Is this plagiarism? Yes? What is it? It is the Gettysburg Address. Exactly. But again, if you put those words into Turnitin or authenticate, is it going to say? So why do we call it plagiarism if the words are different? The concepts are the same. The way they are layered are the same. When are you most apt to plagiarize yourself? Can you plagiarize yourself? How? When are you most likely to do that? How am I doing on time?
I try to publish things in the same area.
Uh-huh.
And so sometimes particular sentences that I'm just stuck to, went to, and then, lo and behold, it's up another script.
So the comment was that sometimes you reuse the same sentences in manuscripts of the same area. Absolutely. And I think, sometimes the Gettysburg Address illustrates that. How many different ways, for example, can you say that the frog is green? Right? I mean, sometimes there are facts that are well accepted. But when are you most likely, again, to plagiarize yourself, aside from those sentences here and there in a paper?
When you don't cite.
When you don't send it, but the methods, right? So you list out how you did your particular experiment, analysis, and so forth. Well, when you publish it the first time, that's fine. But then it's owned by the journal, right? It's copywritten by the journal where it was published. So technically, you are, breaking that copyright if you use it again in another paper that's yours, even though you're the author. So that's difficult, right, to change it up. And journals have different areas of, attention to that. But that's also why you find yourself going down rabbit holes. If you're looking at a particular protocol and you keep following the references back and back and back, right? So that's why. So just to be attentive and to be aware. Yeah.
So which means directly related, since you said journals have copyright.
Mm-hmm.
What if journals are not providing full text free? And what if we upload it to some sites like ResearchGate or anywhere else as a full text? So do we have the right to upload our?
So my understanding so the question was, if you upload this to ResearchGate, for example, excuse me, does that obviate the need for the copyright, or is it a way around the copyright? My understanding, again, of ResearchGate is that it just posts publications that are already, or manuscripts rather, that are already published. I may misunderstand that, but if it's already citing that journal, then the journal has that copyright active. It's protected.
I'm getting a request that, look, full text on it. I keep ignoring it. I'm compelled to keep asking me messages. So I don't know the way around it. Should I just send them, or would I be?
You would probably be violating the copyright. I mean, certainly you could ask and write the journal. That's what I would advise, just to be safe. You don't want to land in hot water. Okay,
Push that.
got it. Yep. Yep. I can try and put it on like this. Okay, so to illustrate how common misconduct is, I've added sort of this timeline almost of comments from different studies that have looked into this. Excuse me. And you can see in effect that it has somewhat grown over time. So here, there we go. About 9% of those surveyed said they had observed direct evidence of the FFP, with only a little over half of that reported. And then in 2015, just a couple of years ago, or a few years ago, 2% admitted to fabricating or falsifying, and 14% said they had observed it in their peers. Again, you're going to get these slides, so if you want to go back and look at the embedded links, you can look at the cases posted through the NIH ORI website, and you'll see UAB's case there in 2009, but it will chronologically give you a list of cases. And now, this is not working. It's not working. I'll let you, yeah. Excuse me.
Is this, I might use this myself. Yeah, it should.
Okay, thank you. So also too on the, there we go, the ORI website, you can actually see numbers or data across several years back. So here you can see in the blue, there we go, the number of allegations reported either directly to the ORI or through their home institution. And of those inquiries, there were very few investigations or those that met the need, bless you, to move forward to an investigation. Again, we'll talk about that process here at UAB in a bit. Of those that went forward for an investigation, thankfully only a fraction were found to actually have misconduct. And of those that did have misconduct, these various cases, most, it's probably not surprising, fell in the realm of falsification and fabrication here. So who do you think was the culprit in most of these cases? Was it the technician? Was it the trainee, the student or postdoc? Was it the professor, the mentor, the principal investigator? Anybody guess? Say again? The PI? So here you go. It's the way they have it here shown. You can see that it's a spectrum. Postdocs have sort of the number on face value, but if you add up all of the faculty, the mentors, that actually exceeds, right? So it's about 35. You add assistant, associate, and professor, as well as principal investigator, which is probably shared among those. It well exceeds that. Why does this happen? Why would misconduct, why would it happen? Why would someone falsify or fabricate? What's the driving force? Say again? Pressure. Pressure for what? Getting funding, getting published, getting promoted, keeping your job, getting a job, right? All of that pressure. So, I mean, we could probably talk for hours. Is that the right incentive to use, or the right metric? That is to say, the number of publications and grant monies for getting or keeping a job. That's a different day. But I think it's important to remember what drives this activity. People aren't doing this because they want to intentionally mislead. I like to believe that. But because they feel a little bit pressured, mostly pressured. So just to keep that in mind as you go throughout your training and work. So we've talked a lot about what is misconduct. What is not misconduct? Okay, so citing the source is the proper thing to do, yep. But what is not misconduct? Okay, here's a hint. We're human.
Any of mistake.
Mistakes, right? Honest mistakes. We transpose numbers. We get things mixed up. You know, we're human. Also, difference of opinion or difference in interpretation, that's not misconduct. That's why we do science. We like to argue. While I was training postdocs and graduate students, if my students weren't arguing with me, I was worried because they weren't doing the reading, they weren't critically looking at their work. That's, you know, having a difference of opinion is how you move that needle and that's when the science happens, right? But that's not misconduct. I want to touch a little bit on rigor and reproducibility. I don't know if this group has gone into this topic in great detail, or how many have worked on or completed the Kaizen. So you're familiar. Okay. This is a new buzz term, certainly in the last couple of years, that NIH is very aware of and conscious to. And again, it's impacting society. Because as was noted, if our work isn't rigorous and reproducible, society is first not going to trust it, hence the whole vaccine issue as an example, or participate in our studies, or even want their tax monies to go to fund our work. So that's an inherent responsibility that we have. And just as an aside, I go back and forth to DC for NIH study section a good bit. And I was flying back with a colleague of mine on the shuttle from DC back to Birmingham. And who was on her flight but Senator Shelby? I hope I run into Senator Jones soon. Senator Shelby was on it. And we were a discussion of where he thought NIH funding was going, because certainly he has invested. And he expressed concern, I'll put it this way, that we weren't being rigorous enough as scientists, and that we needed to up our game. So we have to definitely prove to not only, you know, society writ large, but our own leadership that we really do matter. So again, rigor and reproducibility is now noted and required section in NIH grants, that you do have to provide information about how you're going to make sure you test certain samples and reagents in particular for your work, such that it is reproducible. How many use, in your own work, how many use antibodies or some type of perform basic cult science? Okay, for your antibodies, how do you test them? Do you test them?
Yes. For every experiment, I try to have a point and
Okay.
sure.
So exactly right. To do that positive and negative control, to make sure that the company sent you the antibody and it's detecting what you think it's supposed to be. So that's a great example. How about who does cell culture? So in your cell culture, do you use fetal calf serum? What's in fetal calf serum? All types of things, growth factors, right? It's the secret sauce. When we would do culture and we found the fetal calf serum that worked, again, air quotes, we bought the entire lot. Because who knows what's in that secret sauce. But that's a good example, though, of how an experiment done with that particular lot of serum may well differ from a lab, whether it's at the same university or across the country, that's doing the same experiment with different fetal calf serum. They're both fetal calf, but they're different lot numbers. So that's the challenge that we face, to be reproducible and rigorous, to make sure that those caveats are understood and to be able to explain that to the likes of society and our leadership, to help them understand why things don't always agree. That's science. So I want to touch upon two particular topics next: data management and authorship. So again, we've talked a lot about the FFP and data. What are data, as we discussed? How do we manage that data? So just show this to help you think about all that plays into data management. Who owns the data? Let's start there. The university. The university owns the data, start to finish. Certainly you receive funding from the NIH, for example, to generate that data and to do the experiments. But at the end of the day, the university owns the data. So again, to be mindful and thoughtful about how you collect and store that data. Is it backed up on a secure server? Whether it's observations or numbers, or certainly surveys and tools and things of that nature. Is it stored correctly and securely? Same thing for protection. How long do you keep data? How long do you have to keep it? All those records and such. So the answer, one comment is it depends on if it's animal research or human research. What are some other thoughts? I'm sorry? So whether it's published, okay. How about from a funding perspective? What does the NIH require? Definitely know this. I believe the current standard for the NIH is 3 years of anything that's funded. You have to keep it 3 years beyond the stop of funding of that particular grant. How about from a publication perspective, any data that are published in a manuscript? It's about 7 years. What does UAB require? It's about the same, but here's the hitch. Every time your paper is cited, that clock starts again. So the shorter answer is forever. Where are we going to put all these data?
Is that for UAB or for NIH?
That's everybody. It's everybody. The cloud. How big is that cloud? I guess we're going to find out. But UAB is exploring options to offsite, have secure servers where people can scan and retain data. So, and that becomes really important as we'll talk in a bit about if there is an allegation made, you better be able to find that data. So best practices, careful record keeping, certainly. Electronic records. Does anyone use electronic lab notebook? That too is another area that we're exploring as an option for those who want to use it. There are a number of great platforms out there right now that can help you manage, organize, store your data. Certainly for anybody that does mouse work, it can help you keep track of breeding. That's a pain. But certainly that is something that we're going to explore to help you protect and better manage the data. And again, to make sure that you know the project guidelines and standards for safety and so forth throughout the project. So what are the benefits of good data? Certainly it enables researchers to accurately analyze, to be able to collaborate more easily, you'll be able to find details for the rationale of the project, and certainly it'll help justify to funding agencies. Certainly plays into that rigor and reproducibility as well. Okay, who knows the UAB authorship policy? We have one now. We haven't had it that long, but we have one. What? So you can, and again, you can look this up on the UAB website. It's essentially expectations that any individual as an author has contributed to the work. That should be a given. Doesn't always happen, but certainly has conceptual input and rigor into the project. And these are largely based on the International Committee of Medical Journal Editors, which really facilitates and has the structure of this type of requirement for journals across the board. You may see journals now require that you provide almost like a workflow of each author, what the author did for that particular publication. You sometimes have to provide that. Excuse me. Have you heard of honorary authors? Yeah, what's an honorary author? authorship. Yeah.
They didn't meet this criteria, but you wrote them on.
Right. So about 100 years ago, I did my postdoc at Hopkins, clinical immunology, and we were in our own building, division, and the chief of the division demanded that he have his name on all of the papers that came out. He didn't know me, but he's on my paper. So that's honorary authorship. People that sometimes demand to be on that paper. For example, if you are getting a mouse or some type of reagent or tool, maybe a survey from another group, and they ask or demand and say, quid pro quo, we'll give this to you if you put our name on the publication. That could be a tough conversation, but that's going to be a fine line to determine whether or not that person gets authorship. Just putting that out there for you to think of. Have you heard of predatory journals? Do you get barraged with emails from these journals that have names that are about 10 long? That's the alternative journal, not real, fake journals. Be aware. Just be very wary of these. You can get tripped up. Again, this is an embedded link. You can go to the Predatory Journal site, and you will see this long list of journals that aren't real. Essentially, and I'll list out, in the interest of time here, the different issues with these. They charge rates for publication. Well, all journals do, yes, but there's no peer review here. You can get it published. You can submit it. It'll be published by the afternoon as long as you give them a credit card, right? They target scholars through mass email. They're trying to get you to be on their advisory boards. They may put you on an advisory board anyway. They're trying to validate their existence. I showed up on a couple of these. Make sure you Google yourself to see if you've landed on any of these editorials. Because again, they're trying to use people that have published and are noted in the field to give the perception that they're real. Yes?
They're not peer reviewed. I will assume they're often.
They can sneak into PubMed. I don't think most of them are, but if you were to go on Google Scholar, I bet they're there. I haven't done that recently, but I bet they are, for sure. Yeah?
I got a request to be a reviewer for an article which was in a Predatory. So I was told that since they are letting you peer review someone else's work, so it's probably in the way of getting it a bad idea.
So the comment was that someone has been invited to review for one of these journals. My advice would just be aware, do your background checks on it, and you decide. But don't get caught up in one of these. So we're not going to have time, I don't think, to get into any case studies. But real quickly, to think about case study analysis from an SFNO perspective. Who are the stakeholders, which we touched upon a little bit, society, mainly writ large? What are the facts of any issue? What are the norms? What's a norm? Accepted standard for the field, exactly. And then what are your choices? We can touch upon this real quickly just to get you in that. Well, actually, I'm going to fast forward because I've been talking about the process. So you can definitely go through. I highly recommend going to this interactive case study posted on the Office of Research Integrity website. You can dogleg down different areas and paths by choosing a character and then helping them decide a particular action. But this is a lot of fun. Again, if you're a nerd like me, but it is fun to do. Just want to wrap up and talk about the process. Hopefully this will never happen to you. But if an allegation is made, it starts a process. It will essentially land at the UAB's Office of Research Integrity with Pam Bounellis, who's the officer. And that begins, as I say, a protocol. She'll take this allegation and begin to assess, is there something real, possibly, to to this allegation? Was there misconduct? If she gets through that assessment and deems, there are enough questions here that need to be investigated further, she will convene an inquiry committee of 3 faculty members that have expertise in this particular area. They then will dive through all of the data. Her team will come into the lab. They will gather all the data. Notebooks, hard drives, presentations, grants, publications, even slides from lab meetings. All of that will be essentially gathered up and taken to the admin building. And these 3 faculty members will spend the next 6 months to a year going through all of this material. I've been on one of these committees. It's gut-wrenching to have to do this to one of your colleagues that you've worked with for years. If you get through that process and the inquiry team says, you know what, something did happen and we need to find out how and why, then she convenes a different committee that's an investigation committee. They then will take the next 1 to 2 years, again, looking at all the analysis from the inquiry group and then interviewing people that worked in the lab, collaborators, certainly people off-site potentially, that may have touched upon or been aware of this particular allegation. And then finally, they will have a final report as to whether or not it did happen. If that final report indicates some misconduct did happen, then papers get retracted, money is paid back to the NIH. Certainly it's posted on the ORI website for them to publicize as well. Reporting misconduct can certainly happen to a number of different leadership. If you do have a concern that something is going on, there's a whole host of people that you can turn to as a safe harbor, if you will, whether it's myself or others on this list, or Pam Bonellis directly. Or if you want to be anonymous, you can do that too. I don't know if you've seen these posted around campus, Ethics Matter, but those are resources for you. You can call directly, or you can post online your concern. If you either of these are anonymous, either of these are approaches. For example, the calling routes through a third party, so they don't even take your name. They essentially give you a case number so that you can track this particular allegation. So just one final note before we transition, I take any questions. Well, a few words from Uncle Albert here. Many people say that it's the intellect which makes a great scientist. They're wrong. It's really the character. Again, it gets back to that integrity, doing the right thing when no one's looking. So, any questions? I feel like we've kind of done a speeding through RCR. But again, you're going to get these slides with all these different links for resources and such. All right. Well, thank you all, and I'll turn it over to Carrie. How are you?
Take a 2 or 3 minute break if you need to stretch your legs. So we have a big off-site group who are coming in.
Oh my!
So, yeah, so you need to use the mic.
All right, let me. Where do I plug in first?
You just put.
Just put it on.
Oh.
You're all.
So if any questions come up,
Repeat them.
if you repeat them,
Yes.
paraphrasing at least. And then I'm monitoring a chat box of people offsite. If they have questions, I'll stand up and ask them on their behalf. And otherwise, just the usual.
All right.
Jordan, Martin.
So I'm the chaplain. I'm the faculty coordinator.
It's not just the calibration. So she says in order to have it match the calibration, let's have it, let's do this to this participant, let's do this to this participant. So it's a new rule and we just keep
We'll send you a listing of all the sessions that are
make the
coming
data
up.
match. As long as
So, my understanding is Dr. Hunt's supposed to attend everything. And others, this is
yes.
self-motivated here. There are things that we, so normally people sign up in advance. This is a competitive process.
Awesome.
We're happy to have you. There just are a couple of things where we probably so one of them, it's right that there's not really space to accommodate an extra person in the IRB review sessions now because your room is too small, right?
Well, we've been working with them. Last year we were up there. So, but you know, it used to be that we'd have 3. We can max 2. Just 2.
So that's we've only so we've done it just so nice. Well, every week after the session, I get to go visit an IRB section to see what we're doing inside each. That's how it's all about. Helps you know how to write a good IRB yourself and you get to that point. Unless somebody is expecting to go or so if you really want to do that, you could ask Jim on a wait list for an session of space. We also will have a period where I teach you an abstract and a short talk. We have designated class sessions where people do a short talk, so they're trying to have that. We actually don't have time in our regular schedule for all the people who are.
Just want to get something to drink. Excuse me.
For people who do everything with their
I've got to go. Yeah.
She needs to go. Okay. So anyway, if you can just let Jim know how to reach you, that would be great. Okay. So the second half of today is Carrie Allen, who is the assistant director of the IRB here. The IRB operates out of the administration building and reviews every human studies protocol that's performed by investigators at UAB. Actually, I guess that's not absolutely true anymore because we participate in some group activities where if we're in a cooperative study, an IRB at a different institution may actually be the IRB of record for a study that
We've actually been doing that for a while,
right,
so.
Right. So maybe you can address that a little bit at some point. But anyway, she's one of the main people who really organizes the interactions between the investigators and the institutional review board, and is going to give us a general overview of how IRBs came to be, what they do, and how they operate. Thank you very much.
Hey. All right, so hopefully this won't put you guys to sleep. I find the history of the IRB and human research ethics kind of interesting. Others may not be so fascinated. So I'm going to quote Albert Schweitzer instead of Albert Einstein. So, first step of evolution of ethics is a sense of solidarity with other human beings. I mean, it's just kind of a basic premise that human subjects research is based on, is tuning in to your participant. So, wow, that was something really fast. All right, I'm going to just not use that. I don't know what just happened. but you just saw every slide I'll ever have. Holy crap. We're going to just start over. Look, I was on 33, and we're done. Okay. Let's start here. The basics of institutional review board, which is institutional review board, not investigational review board. A lot of people think that. We have the very basics that the risks to a human subject are minimized, not null, not non-existent, just minimized. There is an inherent risk in almost any research. Most research involves at least a basic risk of loss of confidentiality, and then the risk kind of rises from there. The risks are reasonable and anticipated, and we'll discuss in a minute where that comes from. An interesting part of human subjects research that people don't think about is that the selection of subjects is equitable. What that means is you aren't using vulnerable populations because they're the easiest to get your hands on. You are including people in critical clinical trial research opportunities based on in an equitable fashion. So the history of drug development has there's a long history of excluding pregnant women, of excluding children, of excluding minorities. And as you know, clinical trials can sometimes be the Hail Mary that someone needs to live. And so we want to make sure that we're including people all the way through. In Alabama, one of the bigger issues we have is people who speak Spanish. We have a lot of South American and Central American people who live in Birmingham, and translating consent documents and translating documents from your research is a costly practice. And human subjects regulations, and I'm going to say this brutally, we don't care. It doesn't matter that it's a costly practice. The selection of subjects should be equitable. And then subjects are fully informed of what their participation will entail and the risks and benefits. So as I move through these slides, you're going to see, oh, good Lord, am I just PowerPoint idiot today? So we're going to go through these slides of historical examples of ignoring that last principle. It really starts in the Nazi concentration camps. There were a lot of really horrific experiments that were done during that time on the prisoners. This is one of them. This was the high altitude experiments, where they wanted to find out what happened to their Air Force if they ejected out of a plane at a high altitude. So rather than put their people through that, they put the prisoners through that and stuck them in vacuum chambers and duplicating that air pressure. 40% of them died. I'm going to keep doing this. The cold exposure experiments is very similar. So if they eject and land in the ocean, how long could they survive? So they put prisoners in tubs of ice. They made prisoners drink salt water. It was terrible. They have pictures. They have a lot of documents about it. So ethically speaking, I know you guys just talked about ethics. This is really an interesting ethical issue in human subjects research. I've been to a few national symposiums where this has been discussed because the experiments were actually conducted properly. The experiments they had high power for statistics. They conducted them as true randomized controlled trials in some cases. So how horrific they were and who they used and what happened, the data itself is actually beneficial. So there's a huge and very interesting discussion, and you can go online. It's been a lot of discussions about it. NPR did a great series on it about what to do with this data. We have the data. Do we use it and not put, and not, waste the life of the person who died getting it? Do we not use it and reinforce these terrible techniques? So it's when you really want to get to the heart of ethics, this is a really interesting discussion that's out there, and I find it fascinating. I've sent people to it, but anyway, that's neither here nor there. So after the concentration camps, there was the Nuremberg trial, and all the doctors went on trial. There were 16 defendants, and we know there were lots more. But 16 went on trial, and 7 were hanged, and 9 went to prison. After that, we had the Nuremberg Code. The Nuremberg Code gave us these principles: voluntary, minimized, benefit society. And a lot of people don't realize this last one is that it should be based on preclinical studies in animals. So that's where animal research kind of came from. But this is a code. This was not law. It was just a suggestion from an international board. So Jay Katz, who was an ethicist, said, it should be, that code was good for barbarians. It should be unnecessary for ordinary physicians. Well, history of human subjects research tells us that's not true. That's just not. So we flash forward a little bit, but not that this is the next incident. This is just a prominent one. This was the Jewish Chronic Disease Hospital, Sloan Kettering. They wanted to find out if you could catch cancer. So they injected cancer cells into elderly patients who had dementia. And they told them that they were getting a skin test. Now, we all know you can't catch cancer. And so none of them were actually harmed. But what if you could have got cancer? Then they would have all gotten cancer. Interestingly, 3 years later, one of those guys is named president to the American Association for Cancer Research. So, yay team. Then we have the Milgram study. This is Stanley Milgram in Yale. He has anybody heard of this? It's interesting. I think they made a movie of it recently. So for the few of you who may not have, they brought people in. They had them administer increasing shocks to a participant in another room for not answering questions correctly. So they kept going. The researcher would stand above the guy sitting at the table with the dial and saying, you need to increase that. You need to increase that. And the dial was made so that it was clear to the participant that they were nearing lethal shock areas. And people were screaming in the other room, crying, begging to stop. Please don't do it. Please don't do it. 26 out of 40 gave the maximum shocks. Now, we laugh because we think, holy crap, and that's crazy. Those people had therapy for the rest of their lives. They were messed up. If you read interviews from some of them, they've never recovered from this. Because how would you feel if you found out you were capable, without any pressure whatsoever, without any knowledge of the human, that you were capable of killing someone so easily? It's a really messed up study. It's really messed up. So obviously no one was being harmed. They were co-conspirators on the other side. But this was one of those real ethical protocols that really messed up their participants. It was rough. After that, in 71, we have the Zimbardo study, which is the prisoner study, which is based on the SS concentration. This was, Zimbardo was trying to understand how the SS was able to just implement orders without thinking. So he just built a little mini prison in his basement in Stanford. He got volunteers to come in. I think he paid them $7 a day, and they were students. As they walked in, he'd say, "You're a prisoner, you're a guard, you're a prisoner, you're a guard, you're a prisoner, you're a guard," just random. And the study was supposed to go, I think, for 14 days. It got stopped very soon. 6 days in, the prisoners took their role, the guards took their role, bullying began, food was withheld without any instructions. That the only thing that happened by Zimbardo was, "You're a prisoner, you're a guard." That's it. No other instructions. They weren't told what to do. The prisoners weren't told they can't leave. The doors were not locked. That's the only part of the methodology. You're a prisoner, you're a guard. And during the 6 days that the study did go on, people came through. A lot of investigators at Stanford came through to see the progress of the study. They'd go in and see what was going on. The prisoners would be in fetal positions. The guards would be over there laughing and da-da-da. It wasn't until Zimbardo's girlfriend came through. And she said, "Dude, this is messed up. You've got to stop it." And he stopped it. But it was a really compelling research at how quickly they assumed those roles. And if you'll see down here, Zimbardo testified as an expert witness when the Abu Ghraib prisoner trials were going on. Tuskegee, everybody's heard about that. We're in Alabama. If you haven't, please research it. This was from 1932 to 1972. It would probably still be going on today if it weren't for a New York Times article. As we know, that there have been Tuskegee-like studies that have been unearthed since then. There's the Guatemala study. There have been all kinds of studies like this that have been kind of dug up. I know everybody knows this. This was the purpose of the study was to just look at the lifespan of syphilis, because at the time that the study started, there was no treatment for syphilis. So they just wanted to kind of know what happens. What are the symptoms like? How do they live? What's going on? But very quickly, with 11 years in, they had penicillin, which was a cure, but they didn't use it. They didn't tell any of their participants about it. They actively encouraged participants to sleep with their wives, to infect their wives, and this is documented. They used some very interesting practices in order to gain trust. There's a nurse, and I forget her name, but there's a specific nurse that was responsible for recruiting, and she basically just told lies, but she gained their trust. They never asked about anything. And again, it wasn't until the New York Times article was that this was even considered to stop. If you become researchers in the South, you will still feel the after effects of the Tuskegee syphilis incident. Yes.
I just like to make a comment about it. So I've read Nita's book about it. And the nurse wasn't exactly complicit. She was doing what she was told to do. I don't think she, based on what I read, she deliberately sought to lie to participants. She was given information, and that's what she was carrying out. And it's important to note these were mostly illiterate black men from the black belt of Alabama, and race dynamic. She was black, so they deliberately sent in a black nurse
right
to gain that. So I'm not sure, because, you know, we weren't there. But
I haven't read as many books, so I'm going to defer to you. The book I've read one.
I'm just saying, different accounts, you know, when you're under a microscope, you're willing to say anything.
That's true.
So she may have known, I don't.
I've only read one book, so I'm not gonna count myself.
And also, you had mentioned, since we are on the Tuskegee study, you had mentioned what happened with Jews, but there were documented cases of misconduct of researchers that used slaves, Africans who were brought here in the United States against their will, and conducted all sorts of studies on Africans.
Correct.
To find out what would
Absolutely.
happen.
And
So I just wanted to throw that out there.
this is,
Start with the Jews.
well, this is a good point. This is a history of the human subjects research code. We didn't start paying attention to the abuses in human subjects research until the Nazi concentration camps.
Exactly.
That doesn't mean that was the first instance of abuse. It's just before that, it was acceptable practice or just plain acceptable. We deferred to doctors. We allowed our biases and our prejudices in the Native American population, African American slave population, even through immigration as the Chinese and Irish came through to build the railroads. We have a whole history of abuse in research then as well. We just didn't care. So the first real code of research ethics came from the Nazi concentration camps.
Now, with this Tuskegee, were the participants, did they have sick or getting?
Yes. At the time the study started, there was no cure for syphilis. So this was just just understanding the life cycle of syphilis. This was just, it was a observational study. And it remained an observational study with the, blood draws and everything. It's just that when penicillin was found to be curative for syphilis in 1943, they didn't tell them. They did not treat them. We're looking here. So one thing I wanted to point out is that if you do research in the South today, you will still feel the after effects because it's white coat syndrome. African Americans are still incredibly and justifiably distrustful of research. And so you will experience, this will carry on for decades and decades. Yes.
In all of this situation, where does placebo stand? Like for a layman, it's just a lie. They expect that you're getting a drug or whatever, and instead they're getting a placebo, although they know that there is equal chance that they'll be in the treatment.
That's informed consent.
But it seems like here's solution.
Well, for one, we knew penicillin cured syphilis. In a clinical trial, we don't necessarily know whether the treatment is effective. So, plus we are saying you will be randomly assigned to one of these. One may be more effective than the other. We also require things like data safety monitoring plans. We require in clinical trials, they have to tell us how often they're going to do analysis of the data and what they are going to do if significant findings occur. So we have had studies that have been stopped because the treatment was proven to be effective and they needed to give it. We have had studies that have stopped because it wasn't, or it was detrimental. So we have a lot of monitoring of data now that, so we can tell. But it's all about informed consent. We have very specific language when you are randomized into a study where there is a placebo or a control arm. And that's informed consent. All right. So, spawning from the Tuskegee were the Kennedy hearings. Kennedy hearings resulted in 1974, the National Research Act. This is where the beginnings of actual laws started coming into play. So there was this National Commission for the Protection of Human Subjects of Biomedical and Behavioral Research. And there was a lot of bickering about whether or not to include behavioral. But I think Milgram and Zimbardo helped us with that. So they wrote the Belmont Report. Now, as a nerd of human subjects research, I think the Belmont Report is pretty interesting. It's very elegant. The Constitution of the United States is a very elegant document. It's very short, considering the constitutions of other countries. You know, we have a very small and elegant, straightforward document. The Belmont Report is very much the same. It's written from ethicists' point of view, but also with medical research in mind. And today, it still represents the cornerstone of our industry, and it is as relevant today as any other day. Out of it came three principles: respect for persons, beneficence. So this is 1974, so we still use words like that, and justice. So respect for persons is where our consent process comes into play. Beneficence is the risks and the benefits are known to the best of our ability, are known to the participant. And justice is back to that fairness in distribution, selection of subjects. Again, an issue people don't think a lot about, but they thought about it here, and we continue to struggle to think about it now. But those are our three pillars. From that has spawned research regulations. The last major regulatory medical research, biomedical and behavioral research laws were put into place in 1991. Those are the regulations we're still working on today. 1991 is pre-Facebook, it's pre-internet, it's pre-most technology we deal with today. So as a result, our regulations are actively right now undergoing overhaul. So they were supposed to go into place on the 19th of this month, but there was a 6-month delay. There will probably be a 1-year delay, and there were major changes. But we're still functioning off of the 1991 rules right now from the Office of the Human Subjects Protections. That's our governing body. But then we also have to follow FDA regulations. We have to follow HIPAA regulations. For people who do research on students and student data, there's the Family Educational Rights and Privacy Act, which is basically the HIPAA of educational data. There's the Protection of Pupil Rights Amendments about the kinds of surveys that you can conduct with minor students. DOD has its own, DOJ has its own, Department of Energy has its own. So we have to kind of be aware of a lot of different regulations. These tend to be above and beyond what the OHRP or FDA require. But they're all very jargony and Code of Federal Register and Party of the First Part kind of stuff. But we still understand it as these are our regulations that we work under today. So and they don't always win. Even now, with the new regulations being proposed, the FDA has not proposed new regulations. So if the new regulations had been proposed and gone into effect on the 19th when they were supposed to, then it could be possible that a study that is FDA regulated would have to adhere to old regulations and new regulations simultaneously. And that doesn't sound difficult at all. Yes?
Where can we go to see those changes?
Actually, if you typed in new common rule, our regulations from the OHRP are called the common, that's what we call them, the common rule. If you typed in new common rule into Google, it'll tell you what the new changes are. So let's talk about what an IRB is. So we're required by the code. So we're a law. We are an actual law. We're supposed to protect the rights and welfare of human subjects. We're made up of humans. It's run by humans. It's subjective. This isn't checkbox worthy stuff. The kinds of ethical issues that come to our office are not amenable to checkboxes and simple application of a regulation. There is no regulation that tells me what to do when a parent wants to find out about the whole genome sequencing of their child and tell them the results. Have we diminished the autonomy of that child who may, as an adult, not wanted to know that information? That's not in the code. That stuff comes up every day. So it's made up of humans, it's made up of a board of people to discuss these ethical issues and make decisions just based on the preponderance of evidence and discussion. You'll see that when you come. I hope, sometimes you guys come and you've got, just a boring board of nothing interesting. But then sometimes I'm really excited that some of y'all are there because there's really, a lot of controverted issues and people are discussing and maybe voices raise a little bit, and it's always exciting when that happens. Again, I'm a little bit of a nerd on that. So, per the law, an IRB has to have at least 5 members. Ours has dozens. It has to have at least 1 scientist. Ours has dozens. At least 1 non-scientist. Ours has probably more than a dozen. 1 community member not associated with the institution, and in certain circumstances, a prisoner advocate. So we have all of those. The non-scientists and the community members, they're serving a role. So a lot of times boards, IRBs will get overwhelmed with the scientists on board, and their knowledge tends to drown out the voice of the non-scientists or the community members. So it's a real struggle with IRBs to give these people voice because they represent the participant. They represent the parent of the participant. They're the people who read the consent form and say, I don't understand this. I wouldn't know what you're asking me to do. I don't know what this means for my kid. I don't know what this means for me. So they're a very important eye and a very incredibly important check on informed consent. As we move through a protocol, the more information that you know about the protocol, the less able you are to provide that perspective. So the more I learn about whole genome sequencing, the more I move on the spectrum towards a scientist. I will never be that. But the more that I learn about it as a board member, the less I'm able to perform these tasks. Because you have to imagine what it would be like to have someone just walk into a room and say, this is what I want to do to you. And you have none of that background information. You don't know what a CRISPR is. You don't know about gene splicing. So if you have an informed consent that says, well, we're going to take your DNA and we're going to cut it and replace it with some other stuff, what does that mean? What does that actually mean to me? What are you telling me is happening? And that's really high technology stuff, but it can also be as simple as blood draws. I mean, if I have HIV and I'm going in and your research is, I want 50 mils of blood from you so I can do this other research. Are you asking me to come in so that your research team can draw 50 mils of blood from me? Or are you my doctor and the next time I come in and I have to give blood, are you getting an extra 50 mils? Something as simple as that is the difference between 1 stick or 2 sticks. Now, We don't think of sticks as, high risk, but if you don't like getting your blood drawn, you might want to know, oh, you're piggybacking off of my clinical draw, that's fine, it's just a couple of extra vials, or I don't want to get stuck twice. I don't want to do that. So even small studies like that, studies about data. I'm going to collect a bunch of data from your medical record. Well, am I sharing that with anybody? And if so, what am I sharing? Where is that going? Who's going to have hands on it? All of those things a community member really helps us with. We also have, in the South, we employ we have two boards. So we have a clergy member that serves on each of our boards. We have a Methodist minister and a rabbi. And we feel that given our community and what the South is like, that voice is an important voice. So we include them in our board as well. If we have research that comes across that we don't have the expertise on, we go find the expertise. So we're allowed to bring in people who do understand what CRISPR technology is about and can explain it to us so that we can properly review the protocol. So we have the Belmont report that gives us those basic principles, and then we have the regulations. And then the IRB, they come in with the decisions. Yes, thumbs up, you can do it. No, or you can do it, but make these changes. And then the investigators have to actually enact. Now, we don't have a whole lot of oversight. We do some check-ins from time to time, but we're not there watching them do it. So we do things like this so that we can really impart the importance of ethics and the consequences of bad ethics that can occur, because at this point you're kind of on your own to do what you need to do. We're allowed to approve protocols. We're allowed to require modifications. We can disapprove research. Now, I've been here 9 years. We've done that twice. We're not heavy-handed on that, and most boards aren't, because they'll work with the requiring modifications to get it to an approvable state. The last one I remember was somebody who wanted to pull healthy molars from middle school kids, and we couldn't quite figure out his justification for that. The molars weren't bad and they didn't need to be pulled, so we said no. We can review the modifications, we do audits, and we can suspend due to misconduct, due to information available, and we can terminate. So we do have that power. We're independent. We report to the Office of the Vice President for Research. Chris Brown is our direct report. However, we are by law autonomous. He cannot interfere with our decisions. He can't come in and say, this guy's bringing in $2 million, or hell, that'd be nothing, $14 million, and we really want you to approve that. Can't do it. And we have repercussions if he tried. So we're independent. IACUC is too. The animal version of us is also independent. So we talked about all that. I'm not going to go into that. So really quickly, and I have no idea of my time. How am I doing? Okay. So there's a couple of definitions that I think are kind of important for people to understand when they're learning about the IRB, because as we grew up, as we went to elementary school and middle school and high school, we did research, right? We researched Nietzsche, or we researched the Pilgrims, and we researched all sorts of things. Well, that's lowercase r. You can research how the best ways to fix your faucet in your kitchen. We have a very strict definition of research, and it's a systematic investigation. So that's the first part of it, systematic. That means it has some official design. You're collecting more than one piece or one data point or one piece of information. So it's a systematic investigation that is designed to develop or contribute to generalizable knowledge. So you can do quality improvement that looks and smells a lot like research. You can do all the same activities. You have a problem in your clinic with increased staph infection. So you want to implement an evidence-based protocol from Johns Hopkins about how to reduce your staph infections. And you want to make sure that you, in fact, have reduced them. So you're going to look at data prior to, and you're going to look at infection rates prior to, and you're going to look at infection rates after, and you're going to maybe interview staff to see how they are working with compliance of the protocol. You may even want to have a survey of the patients. All of that is very systematic. But if it's not designed to develop or contribute to generalizable knowledge, if you're just doing all of that to make sure that you have, in fact, reduced your staph infections, and then, yay, you did, cool, that's quality improvement. But if you do all those activities because you're coming up with a new way, you have a suspected new way of solving this problem, and you're looking at it thinking, if this is true and this works, then other small clinics could probably implement this at no cost. I am now contributing to generalizable knowledge, and now suddenly it's research. Same activities. One is research, one isn't. And it has to do with that intent, that second thing. Both have to apply. So if it's not systematic in nature, but it does contribute to generalizable knowledge, it's not research. If it's systematic in nature, but it doesn't contribute to generalizable knowledge, it's not research. And when it's not research, the IRB is not involved. Yes?
You just have to state that you're contributing to generalized research or knowledge, or do you have to?
It normally comes in your purpose. So if your purpose is the Oliver Medical Clinic in Hoover, Alabama is experiencing increased staph infections, and we want to implement this Johns Hopkins protocol to reduce staph infections, and to do so, this is what we're going to do. I read that, I hear quality improvement. If your purpose is and you've provided background on the problem and saying, but no definitive answer on reducing staph infections in rural medical clinics, so we're going to try these things to see if we can reduce, then your purpose is a research purpose. So it's all in how you fashion that research question, and we want to make sure it's clear. Yeah. So that's our big hurdle. Is everything okay? Okay. We apply this question directly as written. It is the first question we ask. So we get a protocol in front of us. The first question we're going to ask is this research? If the answer is no, we're done. So case reports, 3 participants or less, that's not research. You're still gathering data, you're going into the medical record, you're doing all these things, but we don't consider that systematic in nature. Quality improvements, like I talked about. A lot of times people aren't sure, they can call us and we can give you a determination. But a lot of people, you'll hear this, that if you publish it, it's research. That is not true. Published doesn't equal research. A lot of things that are published that are not research. That myth comes from probably the 90s when there were far fewer medical journals to publish in. Now you can't swing a dead cat without hitting some kind of journal to publish in. And there's a lot of quality improvement journals out there that just publish quality improvements. There's case reports. There's all sorts of things that are publishable that are not research. So that is not the litmus test. So then we have human subject. Human subject has lots of moving parts. So the first part is living individual. So Research on whole brains, not research. I'm assuming those people are dead. I'm just going to go out on a limb and say if they don't have a brain, they're dead. Research on homicide rates, depending on what data you're collecting. If you're collecting data on the perpetrators, you might be doing human subjects. But if you are collecting data on the victims, they are not alive. Motor vehicle accidents that result in deaths. We get a lot of that through the emergency department. So living individual is the first part. About whom, so I'm going to collect information about a living individual that is either through an intervention or interaction. So that means I talk to you, I collect blood from you, I poke you, I see you, I email you a survey. That is an interaction. If I email you a link to a survey, I have interacted with you. If I call you on the phone, I have interacted with you. If there's an intervention of any kind, if I give you a drug, if I draw blood from you, if I make you run up and down stairs, if I ask you questions. So, living individual about whom an investigator conducting research obtains data through intervention or interaction, or identifiable private information. Identifiable public information is not human subjects research. Identifiable not private information. We had a study that came before us that actually required some ethical discussion. They wanted to go into a courtroom and just observe the jury through testimony on several criminal trials. They just wanted body language and that sort of thing. We had to make a decision. Is that private? Is that considered private? Private is a place where one can reasonably expect that data is not being collected about you. That generally includes a classroom setting, a medical office. But we determined that no, that's not an expectation in a courtroom. So we did not deem that to be private. But we have to look at all those pieces in determining whether human subjects are involved. And that can be, it can be difficult sometimes. So this is our second question. So the first question is it research? If the answer is no, we stop. No more. But if the answer is yes, it's research, then the next question is, okay, well, but is it human subjects research? And then we apply that, all those pieces. Are they a living individual? Are they interacting or intervening or collecting private identifiable data? So we have a lot of things that are research, but aren't human subjects research. Research on publicly available data sets. There's so many out there that are completely stripped of identifiers that haven't even begun to be mined for all the information we could get out of them. We have a cell line policy, so if you're purchasing cell lines from a commercial vendor, then we don't consider that human subjects research. If, however, you are attempting to create a human cell line, so if you haven't read The Immortal Life of Henrietta Lacks, you should, because A, it's page turning, and B, and don't watch the movie, read the book. I mean, the movie's good, but watch the movie afterward because it's Oprah. You got to watch it. But that cell line policy is based on that. So, HeLa cells, HeLa, Henrietta Lacks. There's a lot of cell lines out there that are based on one human being, and we've got new rules about that. So, not new, but we apply that. So, if it's commercially available, not human subjects. If you're creating it yourself, it is human subjects. And you have to have consent from that person. That covers all the issues from that book. And we have data sets. We have a set of approved data sets that can be used without any IRB approval. Yes, ma'am.
What if a cell line was already gathered from a UAB study? Would that be under approved
No. You would still submit what we call a not human subjects research designation application, and you would have to say, Dr. So-and-so has an approved cell line, and I'm using some of those. And Dr. So-and-so would have to say, yes, she can use my cell lines.
And what is it called, the application?
The designation of a request for not human subjects research designation. Because it's not an approval. You're not getting approved research. You're getting a designation that it either doesn't fit the definition of research or it doesn't fit the definition of human subjects research. So it's a designation. It fits. Okay, so that's important too. We think of research involving people that you interact with, that you get consent from. That really only accounts for about 1/3 of the research that's out there. 2/3 of the research involves people you will never see, you'll never meet, and they'll never know they're involved in research. They'll never know that you looked at their medical records for research purposes. You'll be granted a waiver of consent and a waiver of HIPAA authorization, but you have to meet certain criteria for that to be the case. But the majority of research is done on tissue and data and not involving any kind of interaction or intervention. So when we ask questions like, how many participants do you have? And you're just getting tissue from a biorepository, that question still has an answer. Those tissues represent people. That data represents people. Never, ever lose sight of that. If you have any kind of human-derived material or data, you are doing research on human subjects, possibly. But I mean, those are people, and you need to respect that all the way through, the whole way through. So we talked about what an intervention or interaction is, but it's important to remember that because a lot of people who do online surveys, they don't think that they're intervening or interacting. They think, well, I don't have to answer all these questions. I don't have to worry about consent. If they take the survey, that's consent. Nope. They're human subjects, and you are interacting and are intervening with them. And not everything's medical. So real quick, we have 4 levels of review. Most people do. Most organizations do. So we have that not human subjects research designation that we've been talking about. But then the regulations have a whole area of exempt. And even with the new regulations, they'll have exempt. They'll be broader. There'll be more exemptions. But it's and then there's expedited and full convened. So when you guys come to the board meetings, these are the only ones you're going to be seeing. Those are the most interesting. So there's 4 levels of review. They kind of go in a area of risk. So we talked about human subjects. I'm not going to go through that. Currently, there are 6 levels of exemption. There's an educational research, a survey research, survey of public figures. That one's going away. Research on pre-existing data. That one's broadening. Research and demonstration projects. We've never had one. We probably never will. Taste and food quality. We've never had one. Probably never will either. But Auburn, that has an ag school, does these nonstop. Mississippi State has an agriculture school. Do these nonstop. We don't. So we don't get these. Exempt is not a verb. You are not exempt from IRB review. It means you're exempt from certain regulations in the common rule, but you still have to apply to the IRB. And there are still regulations that have to be met. Every regulation, every one of those categories has its own little set of rules. For instance, you can do a survey of a student if you're doing it under educational research, but if you're interviewing a minor or doing a survey of a minor about what TV shows they like, you can't do that as an exempt research. You can ask them about homework, but you can't ask them about what they do after school. So there's all these little rules of which ones, some are prospective, some are retrospective. Some allow identifiable data, some don't. Expedited also has a bunch of categories, and full board is any research that doesn't meet any of the other three. And it's normally greater than minimal risk, a lot of drug and device studies. And all prisoner research has to be done under full convened board. So if prisoners are considered the most vulnerable population. And you can see from the Zimbardo study why. So we have members on our board who have firsthand knowledge of the prisoner population and how it works. We do a lot of drug court studies here because we have the task program. So we have that prisoner research person. So when you guys come, I'm going to ask a couple of favors. One, our board chair will ask you questions at the beginning. I'll ask you how to say your name if you can't say your name, where you're from, like what department you're from. He will tease gently, which he's known to do. But after that, you're done. So no asking questions during the meeting. You're not part of the board meeting, you're observing. We ask that you refrain from playing on your phone or surfing on your phone. This is something we don't allow a lot of people to do. So this is a real honor that you guys get to sit in there. So try to stay awake and stay off your phones and stay attentive. You'll be given materials ahead of time that you can look at. I think the board meeting is much more interesting if you're familiar at least with what's been going on, what protocols you're going to be reviewing. You'll have to sign a confidentiality agreement, so please refrain from discussing because there are real issues that come up. You may be asked to step outside if one of the protocols we're discussing happens to be something around what you're doing, or if we are talking about compliance issues. So if we have our compliance people come right at the beginning, and so we may be discussing things that may result in reprimands or actions, and we will ask you to step out for those. Don't take it personally. It's just that's out of respect for the person that we're discussing. Any questions? Yes.
I know timing of these sessions will probably vary based on this.
Yes.
But is there a limit? And then if we have to go,
yes. If you have to go, just quietly pack your things and go. You don't need to say, "Excuse me, I've got to go," or anything like that. Just get up and walk out. If you're on call or if your beeper goes off or whatever, get up and leave, and that's fine. You can come back in if you want. Most meetings last a minimum of 2 hours. They start at 9:30. They start at 9:30. We have gone to 2:30. Probably won't during your time, but we have. Most of them will be over around noon-ish, around that time. Wednesdays, every Wednesday. If you cannot go, we'd like as much notice as possible. I understand there might actually be a wait list for this time around, and so as much notice as possible, and then try to reschedule if you can, because it's really interesting. Yes?
Submit a proposal to IRB, how long?
Okay, so under normal circumstances, I would say 10 working days. That hasn't been true for 6 months because we are not living under normal circumstances. And so I think we're going to have to find out what the new normal is going to be. So 6 months ago, we implemented the IRAP. We were the last group to implement IRAP into the system in research administration. It has not been easy, and it has permanently slowed us down. So what we're in the middle of trying to figure out is how much of that slowdown can be worked within the existing personnel we have, and can we shift responsibilities and resources to deal with that? It just slowed everything down. We probably get 100 amendments a week, and before, most of those amendments took about 5 minutes. And at a minimum, every single amendment takes at a minimum 10 to 15 minutes, regardless of its complexity. So that you just compound that and it just gets really bad. So right now, I think most of the staff is actually somewhere around 20 working days. That's when you'll hear from us. So I would, if you've got protocols that, or you know people who have protocols, tell them, get them in advance. Don't be squeaking them in. That's when you'll hear a response from us, and then the replies tend to go faster. So, any other questions? Okay.
Just one thing I should have said already. So next week the topic will be led by Dr. Tom Huddle, who's a faculty member in the Division of General Internal Medicine, really interested in issues around professionalism. It's going to include more discussion of the Belmont report and some of the other principles that underlie our human studies regulations. Jean will send out some links to some materials that it's recommended you read before you come if you can. They're not terribly long, but it's just worth taking a little time to read, so you'll be able to participate in the discussion a little more effectively. So keep an eye out for that.
And he does expect discussion.
Yeah, he will sit and wait for you to speak for a long time.
It's interesting though. It's not about.
Right, okay. Thank you very much.
When will we be able to sign up for attending?
Oh, so Jean, will you, when do we start the meeting?
I'll have to read it.
Okay, so if you bring your calendars next week so that you can sign up for your availability for the IRB visits.
Yes.